Good morning everyone. I'm Lakshmita Jain. I'm practicing in Jesus College, Uti Road. Today I'm going to explain the size exclusion chromatography. In this size exclusion chromatography introduction, principle, instrumentation, application, we will study about that. Firstly, I want to tell about the size exclusion chromatography. Size exclusion chromatography is a technique, separation technique used to separate the solutes based on their size and shape. Now we are going to explain the principle of the size exclusion chromatography. As you see in the screen, size exclusion chromatography is like diffuses like principle holds good. Its solutes may be in larger size or smaller in size or intermediate in size. We can see here in the screen four stages can be seen here. Assume this chromatographic column is a liquid or gas column. It is a aluminium or any column. Column is filled by a packing material that is going to be a stationary phase. This stationary phase is in porous in nature. We can see here porous packing. Porous packing is in a spherical in shape. This porous material should not be responsible for any unwanted absorption of the solute molecules when the solute molecules are moving through the column. In the first column, we can see we injected the sample through the injector. It may be a mixture, the sample containing a variety of mixture. Once introduce the sample molecule to the chromatographic column, the porous of packing material and stationary phase will be having the penetration towards the solute molecule depends upon the size. Porous, it is in the spherical in shape. The solute will be moved freely and those solutes are going to be eluted first. As we see here, the larger molecules will be in the second size separation chromatographic column. The largely solutes are moving rapidly because they do not penetrate and they are excluded, and after larger molecules eluted, then the intermediate solutes will be eluted after the larger molecules. Smaller solutes are eluted at the least. Between larger and smaller solutes, the intermediate solutes are also eluted. If we place the detector at the end of the chromatographic column, we can see or we can get the peak here. This peak is show about the larger solutes and this peak tells about the smaller solutes. And if we get any peak between these two peaks, that is called the intermediate peak. Let us consider this as the chromatographic column. This red color indicates the large particles and this pink color indicates the small particles. The larger particles cannot enter gel and are excluded because larger particles are in excluded and they get diffuses and they do not penetrate. That's why it has less volume to transfer and elute sooner. That's why it's eluted rapidly and fast. The smaller ones can enter gel and have a volume to traverse the elute later because the smaller particles get penetrate and included. That's why it is eluted leastly. As we seen here, the peak of the larger particles and the smaller particles. As we seen in this screen, there are three group of solutes based on size. That blue color indicates the smaller and red color is the intermediate and the yellow color is the larger size. The smaller size contains 10,000 molecular mass and the intermediate solutes contain 30,000 molecular mass and the larger size contains 1 lakh molecular mass. As we seen in this picture, assume a column, the column containing a packing material, then introduce the solutes of mixture into this column, then molecules are segregates. The larger solutes are eluted rapidly and fastly, and then intermediate solutes are eluted. Then last the smaller ones are eluted. If we perform elution process by introducing mobile phase in a chromatographic column, as we see, larger ones are eluted rapidly and intermediate and lastly the smaller ones. Next, as I said before, size exclusion chromatography is a fractionation of molecule based on molecular size. it have two types of gel filtration and gel permeation. Gel filtration is a packing of hydrophilic. It is a water content. It is a high polar species. It is used to separate polar species. And the gel permeation, it is the packing of hydrophobic. It is used to separate the polar species. The polar interaction will be ruled out in the gel permeation. Only non-polar species will be interact in the gel permeation. Size exclusion chromatography, the columns plays an important role. The analytical column will be the 7.5 to 8 millimeter, and the preparative column will be 22 to 25 millimeter. The usual length of these columns will be the 25 to 30, 50 and 60 centimeter. The porous beads, silica based particles, semi-rigid organic gels, copolymers of styrene and divinyl benzene, that is hydrophobic, and porous classes of silica particles. By using these packings will be construct the columns based particles. It is a greater rigidity, greater stability, and stability at high temperature and rapid equilibrium with new solvents. Styrene divinyl benzene gel and hydrophilic gel will be used in this column because the styrene divinyl benzene gel, it is in hydrophobic and used with non-aqueous mobile phase. The hydrophilic gels will be having the sulfonated divinyl benzene because they undergo easily ionization. Let us discuss about the theory of the size exclusion chromatography. We can calculate the total volume with using this equation. Vt is equal to Vg plus Vi plus Vo. Vg indicates the volume of the solid matrix. Vo is the void volume, is the outside of the porous beads, and the Vi means the internal volume, it is the inside the porous. For large molecules, we can calculate with using the formula VE is equal to VO. VE is the addition of the volume and VO is equal to outside of the volume. For molecules of small size, equation is VE is equal to Vi plus VO. VE is the addition of volume and Vi is the inside of the volume plus VO is the outside of the volume. For molecules of intermediate size, VE is equal to VO plus KVI. VE is the addition of volume and VO is equal to VO is the outside of the volume. K is the selectivity constant, and Vi is the inside of the volume. Then we can totally show the equation is equal, K is equal to VE minus VO divided by Vi. That is equal to CS divided by CM. CS indicates the concentration of the solutes in stationary phase, and CM is the concentration of solute in mobile phase. Detectors we use it in this size exclusion chromatography is spectroscopic detectors and refractive index detector. Lastly, we discuss about the application of size exclusion chromatography. It is used widely to the separation of high molecular weight natural product molecules from low molecular weight species and from salts. GPC, gel permeation chromatography, is used to separate homologues and oligomers. Homologues refers to genes or proteins that are similar, and oligomers is a structure or sequence or chain formed by the repeated joining of smaller units or monomer units. And GFC is gel filtration chromatography, is used for fractionation of proteins and other water soluble polymers. And GPC is also used to analyze the molecular weight distribution of organic soluble polymers. And size exclusion chromatography used to purify and analyze synthetic and biological polymers such as protein, polysaccharide and nucleic acid. Thank you.